So first, thanks to all the speakers for really fantastic presentations today. It's a dazzling amount of information that's been presented. So what I'm going to do with this discussion session is answer a couple of questions that came in specific to NIH funding. And then there were some specific questions that I'm going to direct to some of the speakers individually, and then sort of pose some broader questions for all of you to address. So first of all, there was a question about whether or not there will be additional funding opportunities for the centers. So yes, we are the funding opportunity announcements for recompeting the centers and the Data Management Coordinating Center are working their way through the NIH process right now. And I'm hoping that they will be posted soon. It's sort of out of my hands at this point as they get reviewed by the Office of Extramural Research, but those funding opportunities will be coming out soon. NIH works on 5-year cycles, so all of the centers in the Data Coordinating Center will need to submit new applications and recompete for funding. Someone also asked about the low budget for MECFS research. And let me just clarify that NIH does not have budgets for specific diseases other than those that have been specifically appropriated by Congress, like for Alzheimer's disease or autism or HIV/AIDS. So the funding levels are dependent on the grant applications that are received at NIH and those that score well in review and are funded by the various institutes at NIH. So if we were to receive 10 excellent grants in the next round, all 10 would get funded. So it just is very dependent on the grants that are received and those that do well in review. And just one last comment is that there is ongoing continued interest in support for MECFS research across NIH. What I didn't mention earlier is that there's a trans NIH MECFS working group that meets monthly that's chaired by the director of NINDS, Dr. Cora Schetz, where we work together to really help to understand what research is being funded, what needs to be funded, what's happening in the field. And so that's a very active group that continues to work together. So I'm going to now, I think, start with some questions that were specific for Dr. Nath. And I'm hoping, I don't see him, but yes, you're still here, awesome. So one of the questions was about sort of how the individuals were recruited for your study, sort of what were the criteria for allowing individuals in, and sort of what were some of the things that were identified that excluded some individuals from being able to participate in the study?
So we did have pretty stringent criteria which excluded a lot of patients. They had to meet all the various scales. I mean, the criteria that we have, the Canadian criteria, the FUDA criteria, and all the clinical criteria had to be there. In addition to, the most important thing was that they should have had a documented infection. Okay, the Institute of Medicine, that's the one I was thinking. Okay, so they should have had a documented infection, and a lot of them did not. They have other causes for, or they claim they had an infection, but they didn't have any documentation of it. So that excluded a lot of people. And then the other thing is they had to be, should not have had any comorbidities. By comorbidity, I don't mean POTS and that kind of stuff. Those things you consider as part of the syndrome, but not an underlying like Parkinson's disease or cancer or lupus or some other disease like that can then affect the whole thing. So when we did all of those, then we were left with a small subset of individuals. We didn't take severely affected individuals because we wanted to make sure there's no confounding factor of inactivity. So they all had the activity testing done, and they were fairly comparable. So they were relatively active individuals. And then when we brought them in, that's the interesting thing is, so we did all the screening, we we found substantial number of people have other diseases, you know, and they think they have ME/CSF, but there's some other explanation for their disease. So that whittled out our numbers that way.
Could you comment on what other diseases you identified in those individuals?
Yeah, so they're all different things. I mean, one guy had a retroperitoneal cancer. One patient actually had Parkinson's disease. Another patient turned out he has multiple sclerosis. Another person had an inflammatory myositis. We were doing muscle biopsies on all of them, so we found out this guy actually had myositis.
Mm-hmm.
So there were potpourri of diseases. There was no one particular disease.
And I know you're working hard. You and your team are working hard on the data analysis and pulling everything together. Any timeline on when you think all of that data will be?
Everybody wants to know that. I do too. I don't have control on all these other people. I showed you a whole list of things. Everybody's got to produce the data and give it to us. So we keep cracking the whip, but you know, and everybody has lots of competing things to do. But we meet regularly. I think that's the most important thing. All the working groups meet every week or every 2 weeks, depending on where they are, and they're sharing data and analyzing it. So there's a lot of momentum. So I think it'll come together soon.
Great, thank you so much. So Ian, I think, indicated he would answer this question. Can you get ME officially categorized as a neurological and neuromuscular disease in the ICD codes? Ian?
I don't know, sadly, about how these codes are generated. I don't know, maybe Avi, you do.
Yeah. Well, it's Medicare, you know, and so the Social Security Administration is very interested in these kind of things, and they have a big say in this process because they determine disability and all those kinds of things. So they're as far as I know, they're looking into these things. But again, you know, I am no expert in it. In ICD codes, we use them. We don't know how we generate them.
Sorry.
Dr. Bateman, do you want to comment on that?
Yeah, I'm not an expert, but because I worked in private practice and then in a nonprofit, I'm intimately connected to ICD coding. If you're in a big institution, sometimes somebody else does your coding for you, right? Or a lot of it. And initially, it was really intended to help do disease surveillance, right? And partly for Medicare and insurances. But the US follows the WHO ICD recommendation, so they're about a year delayed behind. And so there's a lot of pressure to not rock the boat, kind of, right? Because there's a big established way of doing it and we can make some changes, but it's just a huge, big, complicated institution with many players. And it's quite difficult to get change to happen. But I think that they're hearing the message the last few months. It is a big problem.
So is it generated by WHO or?
WHO has their own recommendations for ICD codes, and then the US does their own, but it's always after and based on what WHO does to some degree.
Yes.
And the problem is most doctors don't pay any attention to coding when they're coding their diagnoses. And even our electronic health record, well, this is a good example, is the CFS code disappeared with ICD-10 coding. It was in ICD-9, but they didn't replace it with something that was appropriate. So it's a very complicated conversation that's too big for this forum here.
Okay, so I'm going to jump to a question for Maureen and then maybe a broader question. Is there any evidence of higher incidence of Epstein-Barr reactivation in ME/CFS patients or in post-exertional malaise?
In our data, we actually don't have evidence for reactivation. That doesn't mean that other people don't. In our particular people that we looked at, our 105 individuals, we didn't see a lot of evidence for reactivation. However, we didn't do every test that one might want to do to determine if there was reactivation. And probably That's a question that Cindy Bateman might want to address.
I'd be happy to give it a try.
Sure.
And
yeah.
I think Ian can add in here too. It's very difficult to capture reactivation.
Hmm.
So reactivation means the cell that the virus comes out of its latent state and starts to replicate. But from what I can understand, it can replicate locally in local tissues and exert an effect which would be very difficult to detect with PCR. And then it can become more systemic. And so I've seen people occasionally have PCR positive EBV or other viruses in certain gold standard workups, but the antibody titers cannot tell you with enough specificity or sensitivity if reactivation is occurring. You have to capture and show the presence of active virus to be able to prove it.
So I have a couple of comments on this. The first is that we've now looked at 3 different large sets of patients who have ME/CFS as well as controls. And when we use PCR, which is the method that's used to detect genetic evidence of the virus being present, there's no difference between cases and controls. If you look at blood samples, which is the only thing that we can look at present, the alternative approach, which is the one I alluded to in my last slides, was to use antibody tests, which is evidence of the virus becoming activated in some part of the body that you can't sample. And we don't yet have that information. So I would hope that within the next 6 to 12 months, we will know the answer to that question. It's a good one because there are several examples I know where we have found evidence of EBV growing in people, and those people have responded to valganciclovir, which is an anti-herpes drug. It's likely to be a small subset of patients. Over.
Can I add something really quick?
Yeah, sure.
A couple of interesting studies I remember emblazoned in my mind is that when astronauts come back from space and land, you can usually detect PCR of herpes viruses in their saliva. So that stressor is enough to trigger reactivation, but not necessarily enough to create a disease state, right? Their immune system handles it. And I've also read a couple of papers about EBV and herpes virus reactivation in really sick patients in the ICU with COVID. So reactivation occurs when the body is very stressed and the immune system cannot control the rate of reactivation. So it's hard to catch, and I think it would really require a stressor, a significant stressor, to be able to determine it.
Yes, I would agree. I mean, the problem with EBV is that you can non-specifically get activated. Everybody's got it. And so it's like on the Neurology Council service, which I oversee. We get EBV positive in the CSF quite often in all kinds of patients with unrelated diseases. We just ignore it because it's positive there, just it gets reactivated and probably subsides. I don't think it really causes any disease. And so trying to figure out when it's actually pathogenic and when it's just a non-specific reactivation is a bit challenging. Same is true of HHV6. That's another herpes virus. I find it activated every year.
Yes, I'm going to switch to talking about mitochondrial dysfunction. You mentioned that, Dr. Nath, about mitochondrial dysfunction in immune cells, and I know Maureen Hansen's group has looked at this as well. So maybe start with you, Dr. Nath, and then ask Dr. Hansen to comment also. Yeah,
so I mean, we've done 2 things. One is we did sequencing of the mitochondrial genome. We didn't really find anything unusual there. Then the second thing we did was we looked at the mitochondrial respiration function. And, the most we did was we took the seahorse instrument and we took whole PBMCs. So they're not fractionated cells, they're actually whole PBMCs. And we found that they moved to anaerobic respiration very quickly. So, so that's really the major finding there in these patients. And if you exercise them, you'll find that your findings get exacerbated. there's a lot more that needs to be done to kind of sort out why is it, where in the pathway it's changing, what cell types are actually affected and which ones are not. So this is just a broad overview of observation that we have at the present time.
Dr. Hansen, do you want to comment?
We've also sequenced mitochondrial genomes of a larger cohort than Avi has access to, and we found no mitochondrial mutations in the patients versus the controls. In fact, the only bad mutation we discovered was in a control and not in a patient. So, and then we have also used the seahorse to look at immune cell metabolism, but there's something that's confused in the literature a little bit, and that is that if you find a difference in mitochondria in immune cells, it doesn't necessarily mean that that's happening in the whole body, because immune cells have specific ways of using energy and specific ways of responding to challenges. So if you see a defective response in an immune cell, which sounds like the NIH group and we have both seen that, it means those immune cells are dysfunctional. It doesn't mean that your muscles are dysfunctional in the same way. And obviously this is something that we might be able to get at by studying metabolism. Those plasma metabolites may give us a clue as to mitochondrial dysfunction, but right now there actually is not a great deal of information about mitochondrial dysfunction in the entire body.
Yeah, so we looked at muscle biopsies also. We didn't find anything there. Yeah, we did the sequencing off of there. We did the histology on them.
Yeah.
Yes, but Avi and Maureen's functional aspects may nonetheless be down. And I think that's what we're also seeing. I mean, as there are mitochondrial pathways that are clearly not working properly. I think you've seen that, Maureen. I've seen that, Avi, you've seen that. So there has to be some effect. And the question is where and how. Maybe it's epigenetic. Maybe there's some other factor. Maybe it's related to cytokines. All of this remains to be teased out. But there's no question but that mitochondrial function is impacted in ME/CFS.
Ian, would
agree?
you tell everybody a little bit about what epigenetics is?
So most people are familiar with genetics, which is looking at the original structure of the DNA. DNA, however, can be modified in various ways. So the genes, even though they may be the same in two people, may be expressed at different levels or in different ways. And there are also a whole host of other factors that can interfere with the way those molecules which are transferred from DNA to make proteins and other structures are also expressed or not expressed. So we have this sort of blueprint, which we all begin with, but that can vary from tissue to tissue. And the way in which that blueprint is used may vary, just as if you have a different builder, you can get a very different result with the same architectural plan.
Thank you. That's really helpful. Anyone else want to make comments about mitochondrial function or dysfunction? So there's a question, and I think probably this was from Dr. Nath's talk about the ME/CFS patients having fairly normal depression and anxiety scores when in fact they have very compromised lives. And why do you think the individuals with ME/CFS don't show potentially more depression, I guess is maybe where this question is going.
Yeah,
Avi, do you want-
I'm sure there are some people who are more depressed. So it could in part be the bias of the population that we selected. And if they had major depression, we excluded them. So we selected individuals who had mild depression or if they were on antidepressants or something, we made sure that they were on stable antidepressants. So it could very well be a bias over study.
Dr. Bateman, did you want to comment on that?
Yeah, I will say that I think for the most part, depression and anxiety are related to chronic illness adaptation. And what we've been seeing in our long COVID community has just dramatically reminded me how hard it is in the beginning stages of illness. And we just had this huge amount of PTSD and anxiety and depression, confusing the whole picture, because the whole world is having that, right? People without long COVID are suffering from more mental health problems during COVID. And the more people adapt to their illness, the more their mental health symptoms resolve, except for the possibly, normal incidents of other more primary mental health problems. It's just my clinical observation.
See, that's a very good point. I mean, the COVID patients have gone through a lot. I mean, they've lost family members, they've lost friends. The devastation is unbelievable. You have close to 1 million people who probably died in the United States. The official numbers are 700,000, unofficial are close to 1 million. And this is the devastation very different than with ME/CFS.
I would say that's not exactly true. It is in magnitude, but every ME/CFS patient goes through that when they are devastated by disease.
Yeah.
So, but it's isolated. What's brought it to our attention is the numbers, right?
Yeah.
And how dramatic it is. And the stressors are huge, but there are ordinary life stressors that our ME/CFS patients doggedly go through before they get sick as well, right? Which is still different than a mental health disorder.
Yeah, no, that's exactly the point I was trying to make.
Yeah, sorry to interrupt.
That's fine.
So I'll ask Dr. Bateman to comment. This is a question we got prior to the meeting regarding the COVID vaccines and whether they're harmful, beneficial to individuals with ME/CFS. Sort of what have you seen and what's your experience?
So I wish we had, better data. We do have a good data for the whole country. There are definitely, among the millions and millions of people who get vaccines, there are people who have adverse reactions to the vaccines. Both Court Johnson on Health Rising and Solve ME/CFS have done some sort of queries to the patient community. And by far, and actually Solve did it with both the long COVID and ME/CFS. So there's really a small percentage of people who get the vaccines who are feeling worse or a lot worse after the vaccines. And it's actually a little higher in long COVID patients than in the ME/CFS population, at least based on that kind of informal, questionnaire online. So, and I will just say also that most of my patients have done better and weathered vaccines with a mast cell activation regimen before they go for their vaccines. And I've sent a couple of severely ill patients for vaccines with just kind of a standard mast cell activation prophylactic. And, we'd need a big study to understand if that was really true or if we're just feeling more confident when we do that. I have at least one patient who relapsed severely from her ME/CFS and POTS after her vaccine, but she also had COVID 8 months before. So it's kind of complicated.
So there are questions, quite a number of questions about treatments. For example, your thoughts on the use of low-dose naltrexone. Are there probiotics that would be helpful for the low butyrate levels, production levels in the microbiome? Are there clinical trials that people can enroll in? So who'd like to take the first stab at any of those questions?
I could do LDN.
All right, go for it.
I've been prescribing low-dose naltrexone for quite a while, and I would say in fibromyalgia patients, pretty typical fibro without all the ME/CFS criteria. If it went to a phase 3 trial, it would be just as good as the FDA-approved drugs. And I participated in all those trials. It's really quite good for pain amplification. I'm not sure about its role in ME/CFS, and I consider it pretty safe. And so if people want to try it, it's safe and not terribly expensive. We really don't know any downside. And yep. So that's just my comment on LDN. We need more trials. I think there's a trial being established in Canada of LDN that should be launching soon that will really help us understand this better.
What about the question of probiotics, use of probiotics? Any thoughts about that? Or what would you use, what could you
So-
take to help supplement?
So Vicky, I answered this in the chat earlier. I really do think we need randomized clinical trials. Although I agree that some of these interventions are not risky or less risky than others, we really do need to support and get clinical trial data.
I agree.
I absolutely agree.
I'd like to comment a little bit about probiotics. So one, all right, can you hear me? I can't. All right. So one of the issues about probiotics is that they're almost all aerobic bacteria because that's how they're acquired. And most of your gut is anaerobic bacteria. So while probiotics might, help, something that is found to be more helpful are those fecal transplants because they can, replace your anaerobic bacteria also. However, we don't know in ME/CFS yet because those clinical trials for the fecal transplants have been held up. There was one in Norway and one in the UK. So I do think we have to know from clinical trials, but I'm a little, like Ian said, we have to be a little wary of probiotics because I found some papers in which people did do clinical trials for various diseases of a probiotic, and it actually made some people worse. So it's not completely innocuous to take a probiotic. And then the other thing I noticed, there are some questions about butyrate and whether you should take butyrate. And that's another thing that is difficult to answer. I will tell you that when we looked at the butyrate amount in our 105 person cohort, we didn't see any difference in the amount of butyrate in the plasma. So the amount of butyrate in your gut could be quite important, as Ian has pointed out, but what's in your plasma isn't different. So if you wanted to improve your gut, butyrate amount, I'm not sure we know how to do that. And, that would be something that would have to be investigated.
Anyone else want to comment there?
I would endorse the statement that both Dr. Hansen and Dr. Lipkin have made about the importance of clinical trials, even if you have a medication that you think is extremely innocuous. Just multiple examples. If you practice medicine for a while, you know that there are so many examples of where we think, all the scientific rationale is very solid, and we should just go ahead and treat patients. And then, you get surprised and humbled to find out that you actually ended up, all your intentions of helping patients, you end up harming them. A good example is TNF antagonists in multiple sclerosis. Huge body of literature showing that TNF is bad. It causes demyelination, this, that, and the other. Animal models show that. In vitro models do that. And then we took a TNF antagonist, and in rheumatoid arthritis, it works. Works in all these other diseases. Gave it to MS patients, relapsed. So now it's contraindicated in MS patients to give TNF antagonists. The same thing with ALS patients, ceftriaxone. A huge body of literature, how glutamate is bad and you can block its transport with it. Animal models show it works, this, that, and the other. Gave it to ALS patients, they got worse. He said, well, it's an innocuous antibiotic. Everybody takes it, what's the problem? Minocycline, another example. It's an antibiotic, everybody takes it for acne. It should be good for you. Give it to MS patients, got worse. Well, there's a huge body of literature showing it blocks MMPs, it blocks the MAP kinase pathway. All these things are bad. They're activated in MS patients, should definitely work. And they're hurting them. So we shouldn't say that these things are innocuous. Let's just give it and see what happens. There's nothing to worry about. There's a lot to worry about. You don't want to hurt patients. You want to help them.
So I'm going to now sort of ask a broader question and sort of go to Dr. Hansen's trunk full of gold. So how do we, with all of this data we now have from all of your groups, plus other groups that are out there doing research on ME/CFS, how do we begin to now sort out what's cause and effect? And how are you thinking about the next steps in your research in particular? So maybe I'll start with Durya with that question.
Yeah, I guess that's the $1 million or $1 billion question. So I think what we're trying to establish right now is, of course, there's a lot of correlations, and the correlations don't mean causations. We know that quite well. But I think what this data is starting to tell us is to generate some new hypotheses about the pathophysiology of the disease, because I think we didn't know what we didn't know. The way we started was, let's look at as many things as possible, because we have some hypotheses, specific hypotheses, but they might be wrong. So we need to analyze the microbiome, metabolism, immune system, and really come up with some unifying denominators or commonalities that could also allow us to ask some new questions. So that's I think the primary goal. And then I think the other thing that's really, really important is to do small interventional trials. I think we don't have a good animal model. I don't think animal models in general are terribly great either. But doing smaller clinical trials with some of these agents that we discussed, but they need to be done properly. And then follow them very, very closely to see actually, if they are reconstituting or changing the biology of the patient, and does that reflect to their symptoms? So it's really sort of setting the framework. To be honest, 5 years ago when we started, I would have guessed all the metabolites that we discovered and how this would link to the immune system. Actually, we're learning a lot of new things about how the immune system is shaped by these metabolism. It's an incredible world that we're just beginning to discover, which will be relevant for lots of other diseases as well. As Maureen mentioned, we're looking at 1,000 metabolites, and hundreds of them we have no idea what they're doing, and it's really trying to figure that out as well. Not to mention all the stuff that bacteria produce that we need to figure out. But I think I'm very hopeful that we're coming to a sort of a framework that we can really build on in the next coming years too. And importantly, use this to stratify subjects. Again, short-term versus long-term disease, male versus females, and so on and so forth, because it's such a heterogeneous condition, and how it differs from long COVID versus Gulf War syndrome biologically, not symptomatic in terms of symptoms. I think these are going to be really important, and I'm hoping that all this data is going to allow us to do that.
Dr. Hansen, I'll turn to you next to answer those questions.
Well, I thought that Durya explained it quite well. What we think is important is to do multi-omics. So, we and other people too have acquired a lot of data, and it's important to analyze it one at a time. So we want to look at metabolites by themselves, by proteins by themselves, mitochondrial metabolism by itself, etc. But what's really important is to cross-correlate. And one thing that's very powerful is to have a lot of data on the same samples. So we think by having a lot of data on the same samples and then doing cross-correlation, we'll be able to identify some important differences. The other thing is that we feel a strength of what we did was by having samples at 4 different times before and after a provocation. When people have different amounts of, say, a gene transcript or a different amount of a metabolite, we can look at the dynamic change that happens during stress and during recovery from a stress, and that helps level out the individual differences that exist between people. So one of the problems in studying any disease is that in addition to the insult that's causing the disease, each person has their own genetics. And so a lot of what people are saying is heterogeneous disease, it's because people are heterogeneous. And I actually think there is that final common pathway, but because people themselves, they are heterogeneous in what they eat, how they exercise, what their stresses are, their environment, and and of course, very importantly, their infection history, their genetics. So that's what makes it very difficult to study any disease, and particularly in this disease where we're studying people who've been sick for a very long time. But I am hopeful that by doing these cross comparisons and before and after a stress, we'll be able to dissect out some, as Duria said, some new hypotheses for that we can then test further.
Yeah, I've had the luxury of seeing some of your more specific data, and I think what's really quite interesting is, and important, is that in your studies, you're able to separate out what is exercise induced versus what is related to the ME/CFS by doing the 2-day exercise and looking across those 4 time points in individuals with ME/CFS and in controls. All right, Ian, would you like to make any comments?
The one thing that I'd like to emphasize is the importance of the data coordinating committee. We can do our best individually looking at data points and findings that we have for our individual studies. By posting our data, the original data, people worldwide can examine the findings and bring to it insights, new analytical processes that we might not have now, or we might have 5 years from now. This is one of the reasons why, with the Hutchins Family Foundation, with the Chronic Fatigue Initiative, we collected samples and we've made them generally available worldwide so that others can test hypotheses, can cross-check and validate or invalidate our findings, and have access to the data points that we've already generated. We've learned, even over the past, I would say, 4 years of the duration of this particular program, that there are new methods coming online all the time that will give us new opportunities to analyze those data. I don't want people to be discouraged, nonetheless, by what we've done to date, because there is a lot of similarity in the findings. And this is telling us that 5 years ago, we didn't know that we were going to get similar results, but many people are finding similar things with the microbiome, with the metabolomic studies, with the proteomic studies. And that kind of reproducibility lets us know that we're going to be able to find answers that are going to be meaningful. Because for a very long time, there were so few people in this field that you never knew if something was a one-off or was something that you could really hang your hat on. And we have a lot of the latter now.
No, thank you for that comment. I think that's critically important. And one of, I think, as you point out, the benefits of the centers and of the research that's been going on, I think one of the things that gets forgotten also is the negative data, and that you've been able to rule some things out in your studies, as well as being able to identify things that replicate across centers, even given the very different protocols and different cohorts of individuals with ME/CFS and controls that have been involved in your studies. So I think, you know, I think long COVID gives us the opportunity, as Dr. Bateman said, to study the very earliest stages of that transition from an acute infection into a more chronic state and into ME/CFS. But it sure did a number on the ability for the centers and actually for all investigators to really accomplish as much as would have been possible without the pandemic. And I'm sure people on the call today realize that the clinical studies were shut down completely and took a long time to come back up again. And many of the labs were shut down for long periods of time. And one of the issues that investigators are dealing with now are supply chain issues. I had someone tell me that they put in an order for a supply and they won't be able to get that supply until April of 2022. So the pandemic has had really a lot of significant impact on the ability to move forward. But having said that, I think it's really promising the work that's come out. So I'll turn to you, Avi, to see if you would like to make any comments. And you're up next, Cindy.
Sorry, I didn't pay as close attention as I should have. I was trying to answer something in the chat box. Can you just read the last part of it?
No, so I guess what's next? What comes next? What do you think how do we come to move this forward to really identifying the causes and what comes next in the research?
So the way, if you saw my first slide, we've designed it as a 3-phase study. First was to do deep phenotyping, identify the potential aspects that we think are abnormal. So, we did a lot of stuff. You don't need to do that in a large population. You need to identify a subset of tests that you think are going to be most informative. Now you do it on a larger population, right? So that allows you to identify, markers that you can use for clinical trials, because that's what you really want. The clinical trial will have to be done on a substantial number of individuals. You can't do 10 patients here and 20 there, unless the effect size is huge, and which I don't think we're going to find. I mean, it's very clear that everybody is finding some immune abnormality. And I think if you look at our data closely, we'll probably come up with things that are quite uniform here. So, and we have a lot of immune modulatory drugs that are available. We can block any kind of T cell or B cell pathway that we want. And so there may be multiple opportunities of modulating them and trying to see how it affects the disease outcome. But currently we're all stuck with clinical endpoints, but we need biological endpoints. I think these biological endpoints are going to come from the kinds of studies that we're doing and then applying them to larger populations.
Great, thank you. And Dr. Bateman, I'll give you the last word here.
So same question.
Same
What's
question.
next? Oh, that's a hard one. I mean, we've survived this and by just being very nimble and flexible and trying to look and see where the opportunities are to move things forward and get involved. I think that, one thing that's been really good for me is reading and reading and reading the literature coming out about long COVID, because things ring true, right, from what we've already experienced for the last 20 or 30 years. We are part of, this hasn't been announced, but I'll just be general, but we're part of the long COVID funding for centers as a small part with people here in the West, the University of Utah and Colorado and New Mexico. So we're going to keep our foot in the door and keep being a voice. And then I'm looking forward to see what happens from those 40 patients that we enrolled, right, in with Daria and Ian, to see what kind of commonalities we can see between long COVID and ME/CFS. And then there's always bigger long COVID data out there, right, for validation.
So thank you all so much. This has really been a fantastic afternoon, fantastic panel. I thank all of you for your presentations, for your wise thoughts, and mostly for your dedication to research and clinical care on ME/CFS. So thank you all very much. And I'm just going to share my screen here quickly.
And you too, Vicki.
Thank you. So this is a word cloud that I actually made while we had our 2 days of meetings with all of the investigators. I think there were, over the course of 2 days, about 150 people, including patient advocates, who joined us for the center's meeting in early October. And I just started adding in words as people were talking about various different processes, different systems, different things that they're looking at. And I'm just putting this up here by way of, I think, confirming what you just heard, that there's lots of new clues, lots of new evidence, lots of coming together of groups finding similar findings in different cohorts of individuals with ME/CFS that I think really should give all of us hope and the promise that the research is really moving forward, that we do know a lot more now than we did 4 years ago when the center started and before the NIH intramural study. So I know that there is certainly the desire for treatments, for clinical trials. NIH is open to supporting clinical trials. We just need to work with investigators that can help us to put those clinical trials in place, because it's not something that we as program staff do. We can help to support and fund clinical trials, but we need the investigators out there to really come to us with ideas for clinical trials that can be supported by NIH. And so with that, I'm going to end and turn it back to Phil and just thank all of you for your attention this afternoon. Once again, thank all of the speakers and thank the Massachusetts ME/CFS organization for putting this together. Thank you very much.
Thank you, Vicki, and thanks to the organizers. This was
Okay,
a very
well,
good discussion.
thank you, Dr. Whittemore. Thank you, all the speakers. I won't reiterate what Vicki said. She said it quite well. We are just so grateful for all of the work that you do and for taking the time today to share it with us. The conversation has changed quite a bit. Personally, I was diagnosed in 1986. The conversation for decades was, does this exist and is it in your head? I think you've clearly demonstrated we're way beyond that. And the conversation now is this is dreadful and how do we help? And you've clearly demonstrated that's not an easy problem to solve. So we are grateful that you're working hard to make it happen. To all of our speakers, again, to our members who keep us afloat, to everybody who has joined us today, thank you so much for being with us. A reminder that the video of this will be posted probably not until December, but you will get an email sending you to our YouTube site. We will be on for those who want to stay and chat. We will stay on for another 10 minutes with some music. So feel free to enjoy the music and chat away. Thank you all.